This is an overview of electron transport oxidative phosphorylation. Again, this occurs in the mitochondria. Uh, all the proteins involved in the movement of electrons from NADH onto oxygen and production of ATP are membrane bound. There are complexes in the inner membrane. What happens, the purpose of this pathway is to regenerate NADH, FADH2, that's produced in glycolysis and also TCA cycle, two forms, NAD and FAD, that can be used to continue the oxidation of molecules in those pathways. As we reoxidize NADH and FADH2, that process is exergonic. Eventually we pass electrons from those electron carriers on to dioxygen, which is an excellent oxidizing agent, obviously, and in the process liberating sufficient energy to drive ATP synthesis. The coupling of oxidation and phosphorylation occurs not by the formation of like some high energy chemical intermediate, but rather the energy released on oxidation of NADH and FADH2 is captured in the form of a proton gradient, which then can drive, in an exergonic process, the synthesis of ATP. Here are the protein complexes: complex 1, 2, 3, 4, 5. They're very complicated. They're all resident in the inner membrane. NADH and FAD and FADH2 are in the matrix of the mitochondria. What happens is NADH passes its electrons along to complex 1. In the downhill process, there's a lot of iron sulfur clusters in here, ultimately to a mobile electron carrier called ubiquinone. That's shown here. In fact, that's a recurrent theme in this whole process. Electrons are passed from mobile carriers through these protein complexes to other mobile carriers and on down to O2. After ubiquinone, which is lipophilic, sits in the membrane, picks up the electrons, it's reduced and becomes ubiquinone reduced, UQH2 typically. That ultimately passes its electrons on through complex 3 to a protein called cytochrome C, and then in complex 4, cytochrome C passes its electrons on to dioxygen. So in this diagram, the mobile electron carriers are outlined in red boxes here: NADH, FADH2. Ubiquinone happens to be a lipophilic variant that's in the membrane. Cytochrome C is actually a protein that is soluble in aqueous solution. It's actually in the inner membrane space, and of course dioxygen. So those are the mobile carriers. The fact that these membrane complexes are in the membrane is important because that's the only way we could vectorially discharge protons across the membrane as we oxidize molecules like NADH and ubiquinone. There's enough energy in the oxidation of those as we pass them down to better and better electron carriers or oxidants to drive protons across the membrane from the inner membrane, from the matrix into the inner membrane space, and that's shown here through complex 1, complex 3, complex 4. We generate sufficient protons that when they collapse through complex 5, which is the ATP synthase, we can drive ATP synthesis. FAD is used in the mitochondria TCA cycle. As we take succinate to fumarate, you might want to review that. The FADH2 that's generated in the mitochondria can shuttle its electrons onto ubiquinone. In that process, however, there's insufficient energy and no protons pumped across the membrane to actually contribute to the proton gradient across the membrane. So again, just to review quickly, we have a series of mobile electron carriers, mostly hydrophilic, some hydrophobic, that shuttle back and forth between these membrane complexes that are arranged in almost a supramolecular structure, allowing the downhill thermodynamically favored transfer of electrons to dioxygen, and coupling that to proton gradient formation, and then ultimately ATP synthesis. I'd like to briefly show what complex 1 looks like. It's a complicated membrane with a membrane domain and this domain that's obviously poking into the matrix of the mitochondria. This is the matrix domain of F0 of complex 1. And what you can see in here, that there's a series of iron sulfur clusters. I can turn them on and off, or actually illustrate them in space fill. And then there's also a flavin molecule, which is shown in the red. What happens is NAD H dishes off its electrons to the flavin, which is a flavin mononucleotide, and again that process can occur in a two-electron step. And then there's a transfer of electrons through these iron sulfur clusters all the way up to this domain here, where there's a terminal iron sulfur cluster where ubiquinone can then pick up the electrons, and the process is then complete. What's not shown here is the membrane domain for this particular protein. Here is the complex 1 in its entirety, and here you can see the transmembrane domain. Membrane domain through which protons are transferred across the membrane. Those residues highlighted in here as sort of spaceful are involved in the terminal iron sulfur cluster, which is involved in the vectorial discharge of the protons across the membrane. These particular, you can see there's a lot of helices here, and all these particular units here are essentially have homology to proton antiporters, so their function clearly is involved in transporting protons across the membrane. And now you can see I've rendered the transmembrane domain as alpha helices, and I think you can see they're generally oriented in the membrane going across the membrane. There's this one long helix there that seems to be involved in conformational changes involved in proton transport across the membrane. I will review the other complexes briefly in another video.